Next, let's move on to the other topic. And from Samsung Biologics, we will invite Dr. Tam Dong-soo, and he'll be talking of vaccine.
He's going to tell us about the vaccine trend at the corona pandemic situation. She's going to give us the comprehensive perspective. Let me briefly introduce Dr. Kang Dong-soo, and in the Texas Health Science Center and the Thomas Jefferson University. He got the PhD and he worked for various organizations in the U.S. and he is currently working as a head of the biotesting service in the Samsung Biologics in Korea. Dr. Kang, please. Good afternoon. Vaccine trend at current pandemic situation, recombinant protein, nucleic acid, and viral vector. This is the title of my session. My name is Kang Dong-soo, who is working as a CRO of the Biosafety Testing Service in Samsung Biologics. First of all, thank you very much for giving me this opportunity. Thank you, the organizers. Thank you very much. And today's session, I'm going to give you the general information so that we can think about the new vaccine trend. So I collected some journal level information and I collected some of the public information. And for the reference of the information, you can see the source of the information at the bottom of each slide. As for the COVID-19 pandemic, everyone is directly and indirectly affected. As of last weekend, across the world, more than 23 million people have been infected. Already, more than 800,000 people died, and more than 200,000 newly infected cases are happening on a daily basis. To overcome the COVID-19, we needed to develop the vaccine, and we can have the traditional vaccine. However, other than the traditional approach, I would like to tell you what kind of novel approaches are being taken. Before I would like to introduce the vaccine methodology, I'd like to tell you about the history. The father of the modern vaccine, Edward Jenner, Louis Pasteur, Jonas Salk, and Albert Sabin, and another vaccine visionary, Dr. Maurice Hillman. I'd like to briefly introduce his achievement, and then I'm going to tell you more. There are 8 strains of the vaccines that are currently used, including that he created 40 animal and human vaccines. And his daughter was suffering from the epidemic perititis or the mump. And he separated the mump virus and the cultured the mump virus and developed the mump virus. And one of the essential vaccine is the MMR, targeting the measles, mumps, and the rubella. The vaccine has these 3 indications. He's the one who developed the MMR vaccine. He worked for the Merck for a long time and he retired from the Merck. And the Merck vaccine facility located in the North Carolina named after him. Robert Geller, who first discovered the HIV, said that Dr. Hillman contributed to the benefit of the human health, and he is one of the most successful vaccinists in history. Maurice Hillman developed the so-called the first generation or the traditional vaccine. It is culturing the virus and purifying the virus after the attenuation or the inactivation is going to be directly injected into the human body so that the human body can generate the antibody against the virus. The efficacy is really high. However, it takes a long time to prove its safety. Let me tell you a little bit more about the second generation vaccine. The virus will be degraded chemically. It's going to be inactivated, or the gene recombination will be used. And by doing so, people can purify the antigen-specific protein on it, and it's going to be injected as a vaccine. The third generation vaccine is a little bit different. It is not producing the antigen protein. It is helping our body to produce the protein. So we are gonna inject the DNA or RNA that contains specific genetic information. WHO started the Disease Access Program Disease X program is targeting the unknown disease because the unknown disease can cause a global pandemic, just like a COVID-19. Under that assumption, WHO started the Disease X program, including the vaccine manufacturing. The program was initiated in 2018. This is making a poor preparation for the future pandemic, and this is a great reminder because we are currently suffering from the COVID-19. To meet the goal of the Disease X program, treatment and the vaccines need to be developed, and to do that, we need continuous R&D and information sharing. And also, we needed to have the vaccine manufacturing platform or the treatment and manufacturing platform to tackle any kind of a disease X. And also, we needed to have the manufacturing system that can enable quick development and production. Also, we need the environment where accelerated clinical trials are possible. However, what matters most is the safety of the vaccine and then treatment. However, the manufacturing process should be safe enough because when it comes to any kind of medicine, patient safety should come first all the time. And we hear lots about the second generation and the third generation vaccine. Let me tell you about the mechanism of action for those new generation vaccines. COVID-19 can be infectious because the S protein, the spike protein, is recognizing the antibody on the ACE2. So there is a binding between the S protein and the ACE2. As you can see on the bottom right, the S protein, if there is an antibody that can recognize the S protein, and probably the cell infection might be prevented because the binding between the two protein can be blocked. So we needed to have purified S protein directly injected to the human body, or we can encourage the human body or induce the human body to produce the S protein so that the antibody can recognize the S protein. Let me tell you about the recombinant protein vaccine. And it is using the recombinant technology just like the monoclonal antibody vaccine. Only S proteins will be generated, separated, and purified. As you can see at the bottom, you can see the recombinant S protein vaccine, which was produced by the US company M. Insect cell virus, baculovirus, which contains the S protein genetic information, will be generated to infect the insect cell and to culture and purify lots of the S protein. Before moving on to the third generation vaccine, let me tell you about the central dogma, which describes how protein is generated. Because the third generation vaccine is not producing the protein and injecting the protein into the human body, but rather human body is used as a factory to generate the S protein. Cell is composed of the nucleus where DNA is located, and also it is composed of the cytosol where nutrient for cell life is generated, protein synthesis and the metabolism take place. Within the nucleus, DNA can be replicated if necessary, and the messenger RNA, which makes a protein, is synthesized. And the synthesized messenger RNA goes to cytosol and utilize cytosol as a manufacturing factory to synthesize protein. Messenger RNA vaccine is one of the third generation vaccine. It looks like this. The gene of interest where S protein genetic information is located, as you can see on the picture, it is GOI. And also there is a cap, which delays the natural degradation of the mRNA and helps protein generation. And also there is a UTS, untranslational region, and it has a poly A tail. And also the mRNA is delivered to cytosol along with the nanoparticle in order to produce S protein. And at the bottom, you can see the messenger RNA vaccine, which is developed by the company M. And the biggest benefit is it is a flexible platform which meets the need to disease X. When the new vaccine is required, only antigen information is changed and the shutter system can be applicable. Next, let me tell you a little bit more about the viral vector. I will take the example of the adenovirus, which has been used for the targeted cancer treatment or for the genetic treatment. Adenovirus is known as the cause of the seasonal flu. From the genetic information of the adenovirus, we can remove harmful factor and insert the genetic information of the spike protein. After the mass production is made, then the separated and purified virus is used as a vaccine. As you can see on the right, as this is a virus itself, it can easily infect the cell, and the infected cell will produce S protein, and the human body creates antibody against the S protein. In a nutshell, this adenovirus is a delivery tool for the genetic factors. It works as a gene carrier. To sum up the clinical outcomes of the COVID-19 vaccine, in a nutshell, I can say in the case of the second and third generation vaccines, it seems short-term safety is already secured. They are either in phase 3 of the clinical trial or plan to begin phase 3. Convalescent is a blood sample of the patient who recovered from COVID-19, which has COVID-19 antibodies. When we compare the antibody generation against the S protein, I can say its effectiveness was quite proven as a next generation vaccine. Before the vaccine or treatment becomes available, we will be exposed to the COVID-19 and we needed to protect ourselves. WHO or the CDC say just by washing your hand, you can reduce the 50% of the pathogen infection. Sometimes we put our hand to our face 500 times within a single day. When you are wearing a mask, you can reduce the 80% of the infection. And when we have the social distancing, probably it's gonna be the part of our daily lives. And I just told you everything that I prepared under this situation. There are many people who took time online and offline. Thank you very much for joining us. And if you have any questions, please email me and please ask a question. Thank you very much. Dr. Kang, thank you very much. He's from the Samsung Biologics and he is the CRO. He showcased the development process and the history of the vaccine development. I was able to learn a lot from what he said. And there are lots of the vaccine which are in the phase 3 or about to get into the phase 3. It is rather promising. However, at the same time, there will be continuous mutation. So the vaccine development is really difficult and challenging at the same time. Thank you very much, Dr. Kang, for the wonderful introduction.